Good afternoon, everyone, and thank you for joining us for today's webinar. My name is Josh Vickers, and I'm with RTI International's Forensic Technology Center of Excellence. Today's webinar is entitled Emerging Forensic Research, the Forensic Toxicology and Drugs. This is the fourth webinar in our Forensic Research webinar series. We do have four presenters today: Dr. DiCaprio, Mandy Moore, Dr. Carl Wolf, and Dr. Chris Mulligan. We're going to jump right into it. Our first presenter of the day is Dr. Anthony DiCaprio. We will let his intro slides pop up. He will be presenting on development and validation of a blood protein modification assay for retrospective detection and abused drug exposure. Dr. DiCaprio is a toxicologist with experience in neurotoxicology and the analysis of drugs and other xenobiotics in human specimens. Prior to joining FIU in 2009, he served in research positions at the New York State Department of Health, University of Albany, and UMass Amherst. He currently directs the FIU Forensic and Analytical Toxicology Facility, which provides research support to investigators in analytical and clinical forensic toxicology. Let's just jump real quick back to our quick disclaimer. The opinions, findings, and conclusions or recommendations expressed in this presentation are those of the presenters and do not necessarily reflect those of the National Department of Justice. Any products and manufacturers discussed are presented for informational purposes only and do not constitute product approval or endorsement by the U.S. Department of Justice. And now, without any further ado, I would like to go ahead and hand the microphone and floor over to our first presenter, Dr. Anthony DiCaprio.
Okay. Thank you, Josh. I hope everyone can hear me. Welcome, everyone, and thanks to RTI for the invitation to present. So I won't reread my title, but as you can see here, we have an interest in retrospective drug exposure detection, focusing on drugs of abuse. And without further ado, I will begin. So I won't reread this disclaimer, but obviously a NIJ-supported project. So let's talk briefly about what we call the drug detection window. I'm sure most people in this audience have seen something similar to this, where we can utilize various matrices, human matrices, to detect drugs. And they vary quite dramatically in their useful time period within which one can detect a drug. And so we can see from this list of the most common matrices available, certainly blood has a relatively short window, minutes to hours, depending on the metabolic rate of the drug in question. Exhaled breath, another fairly short window. Oral fluid, a little bit longer. Urine is quite variable, of course, with certain compounds being able to be detected upwards of weeks. Of course, that's a very limited number of compounds, such as THC, which are fat-soluble, stored in fat, and slowly released back into the blood, then metabolized and collected in urine. Sweat is sort of a unique matrix in that it's really more of a cumulative dosimeter, because you have to wear a patch in order to collect sweat and then analyze for drugs. Meconium, a highly specialized type of matrix, and that leaves us with hair. Forensic hair analysis, which, as everyone probably knows, can give us much longer time periods of detection, in theory outwards of years. There is a lag period for hair where, of course, the drug or metabolite has to be physically incorporated into the hair, and that takes some time, a matter of days. So hair is really the only matrix that we can currently utilize for retrospective drug detection. Retrospective meaning going back in the past to look for either episodic past exposure or measure cumulative exposure over long periods of time. So forensic hair analysis is fairly well developed, and it's a major advantage. It's non-invasive, and it can produce a map whereby, by sampling segments of hair, one can sometimes interpret the time of an acute exposure. This hair, of course, also suffers from some major difficulties. Everyone is different in terms of their composition of hair. Some people don't have head hair to sample. A major problem is external contamination, and interpretation is also can be a big problem. So we're proposing an alternative to this by means of analyzing modifications, and by that I mean covalent stable modifications of blood proteins. This is not a new technology. It's been used fairly widely in epidemiological studies of environmental agents and occupational, but very, very little application to measuring drug of abuse exposure. Advantages are, I'll show you in an upcoming slide, the kinetics of these proteins. The sample matrix is very homogeneous, meaning it does not vary much between people. We all have essentially the same hemoglobin and albumin, and we can, in theory, assess both episodic and cumulative exposure. Unfortunately, yes, you have to stick somebody, so it's an invasive collection, and the big challenge is analysis. We're looking for fairly low levels of modifications, maybe 1 in a million or less adducts per molecule of protein. And also, we need to get into the area of proteomic analysis, which is not all that familiar to most forensic scientists. Okay, so just to summarize, a typical drug or xenobiotic gets metabolized. One pathway is to an inactive metabolite, which gets excreted. Second pathway is that the drug can be activated to something toxic or pharmacologically active, and then ultimately be excreted. And the third pathway is the one we're interested in, where every drug can be metabolized, at least to a small fraction, into a reactive electrophilic metabolite, which then can interact covalently with either DNA or protein. And we can measure these as markers of exposure. So I'm going to talk about protein adduct, protein modification measurement. Okay, so very briefly, kinetics of blood proteins, and we're most interested in hemoglobin and serum albumin, since they are by far the major protein components of blood. Hemoglobin, of course, is the oxygen-carrying molecule, manufactured in bone marrow, incorporated into red blood cells, and then out into the circulation. This shows the elimination or loss kinetics of both proteins. Hemoglobin really is lost in a zero-order manner with an elimination lifetime of about, on average, 126 days in humans, whereas serum albumin is shorter, and it actually has a true half-life because it's an exponential decline of 20 days, but one can still, in theory, measure modifications out to 40 or 50 days. So these are fairly long-lived proteins, in particular hemoglobin. Okay? So this just is some of our initial work in this field, where we observed modifications in both glutathione, which is a small peptide, circulating peptide, and also proteins from blood, showing conversion of cocaine into a reactive epoxide, which can then adduct the proteins. Okay, so in this project we have a number of stages. I won't go through all of these in detail. The first step is an in vitro approach, just to confirm that we can actually have protein modifications and peptide modifications by a series of reactive metabolites of these drugs in vitro. The next step is to try and maximize sensitivity of this detection, since it is a challenge. These are very, very low levels of modifications. And then ultimately develop a targeted triple quad method that could be used by forensic laboratories. And then, of course, confirm this with authentic whole blood specimens. So the drugs under study in our project are shown here. We have a series of 16 drugs selected for various reasons, variations in chemical structure. We wanted to look at a range of different chemical structures, a range of both illicit and licit drugs that might be observed in drug users, for example, including 2 positive controls, acetaminophen, Tylenol, and clozapine, which have been widely studied in the literature for their ability to modify protein covalently. Okay, so in terms of targets, what exactly are we looking for? Well, we're focusing on free thiols as the nucleophile in these reactions. So one of our models is glutathione, which is a circulating peptide present in very high concentrations in people up to 10 millimolar. It has a function as a protective molecule to actually intercept electrophilic species, which can be toxic, and thus detoxify them. We also have a synthetic hexapeptide, which is shown in the upper right, which also has a single free thiol as a target. And then, of course, albumin and hemoglobin. On the lower left is hemoglobin, and you'll see I've listed a peptide there with the reactive 93-cis cysteine moiety, which is well known as a reactive site in hemoglobin. That is our target for that protein. And in serum albumin, the single free thiol at cysteine position 34. So those peptides that are shown there are actually tryptic peptides. They're generated by tryptic digestion of these proteins, and we can isolate and characterize those target peptides. Okay, so let's move on to look at some of our in vitro work with glutathione and peptides. So these slides will be available to everyone, so I won't spend a whole lot of time, but this is just our trapping assay where we mix drug, human liver microsomes as the source of metabolic potential, NADPH, and a regeneration system, so we have everything that we need to create an in vitro reaction system. We incubate for a certain amount of time. We add our target, in this case either glutathione or the model peptide, and then we do LC-MS analysis. So our strategy is initially to start with triple quad LC-MS on a C18 column. We look specifically for product ion scan for M over Z 272, which is a glutathione fragment that is widely used as a marker for glutathione, and then we compare these to a theoretical list of possible adducts and metabolites. And then ultimately we confirm this by high-res QTOF-MS. So glutathione has the advantage that it's very well characterized in terms of its ionization in both positive and negative mode, and we have a series of characteristic ions. The ions shown here in green, if they are present, confirm that you indeed do have a glutathione modification. The ions shown in red here will actually tell you these are the drug-specific adducts. They will tell you, or ions. They will tell you that yes, we can infer structure of the adduct from these ions shown in red. And finally, by looking at ratios of some of these characteristic ions, we can identify potential sites of the linkage. If you see there on the right, bottom right, we have glutathione, either aliphatic, aromatic, or benzylic linkages. So we can get a lot of information from these studies. To summarize, of our list of drugs, we found 10 out of the 16 drugs were positive for glutathione covalent modification. On the right-hand column, if you see none, that means this is a novel. No one has reported covalent modification of glutathione by these particular drugs. So, example, DZP, diazepam, and the ones at the bottom, naltrexone, oxycodone, and THC in particular, we detected adducts, and that had not previously been reported. So let me just quickly run through a few of these spectra. So these are high-res mass spectra of these various glutathione modifications with the adducts or the ions in color, as I explained earlier. The blue ions to the far right are the molecular ions, so they can give us obviously information about the overall structure. But the ions in red are really the ones that are diagnostic for structure, and you can see MDPV, two different adducts here, both of them with loss of the pyrrolidine ring. And you'll also note that these are metabolites. So we have the demethylation, cleavage of the methylene dioxy ring. So these are actually secondary metabolites that have then gone on to modify glutathione. Same thing with naltrexone, two different modifications. You can see on the right-hand panels where the SG, the glutathione, where we believe the linkage occurs. With the top, it's an aromatic bond. With the bottom one, it's an aliphatic modification. Oxycodone, two more adducts. The oxycodone 2, we were not able to identify exactly what that structure is. And maybe most interesting are the THC modifications. No one has previously reported covalent modification of protein or peptides by THC. And if you look at the THC1, this is actually the 11-hydroxy metabolite, which then goes on and modifies glutathione. The bottom one is the 11-carboxy metabolite, which is bound to GSH. Okay, so one slide here, our model peptide, our hexapeptide. Same idea here we've shown. In this case, it's methamphetamine, showing in vitro stoichiometric conversion of this peptide. So interestingly, we only saw 10 of the 16 drugs adduct glutathione, but all 16 of the drugs were able to modify this synthetic peptide. Okay, let's move on to our human hemoglobin in vitro studies. Again, this is our incubation model. It's similar. The first part of this is similar to the peptides, except that we substitute hemoglobin in place of glutathione, but the first part of this is the same. And we then isolate the whole protein hemoglobin, which we can proceed to do whole protein mass spec on. Alternatively, we can continue on with a triptych digest to chop it up and get our unique peptides to look at. So this just shows an example of what we do in the laboratory, with the top panel showing whole protein analysis, where we're simply looking for an incremental increase in mass of the whole protein, which could be indicative of binding of a single molecule of the drug. And in this case, it's actually an aromatic amine compound that we're using as a reactive compound, just to show the principle of this analysis. The bottom shows a typical triptych digest, and here we have our hemoglobin peptide that contains the single reactive beta-93 thiol. And of course, I don't have time to go through this, but this is the analysis that's necessary. We look for these B and Y peptide ions and confirm location and structure of the adduct. Okay. So in the interest of time, I'm going to skip this. There's a fair amount of software analysis involved in finding these adducts and characterizing them in these triptych digests. And this is what a triptych peptide map looks like, and where we the red enclosed bars there show suspected peptide modifications, which we then go on and do high-res mass spec and high-res MS analysis. So this is a summary of our results with hemoglobin to date, showing positive adduction findings for the drugs that you see in the second column there: acetaminophen alpha PVP, methamphetamine, naltrexone, oxycodone, and THC. along with their peptide masses. The ones in green we actually have MS data on, and I just want to quickly go through this. Next slide just shows what's involved in these analyses. It's fairly complex, and in my last slide I'll talk about what we plan to do to make this simpler. Just an illustration, this is the acetaminophen adduct bound to the beta-93 thiol of hemoglobin and also the beta-112. Hemoglobin has 3 free thiols, which may be targets, with the 93 being the most reactive. Next slide shows adducts for methamphetamine and THC, again at the hemoglobin beta-3 and alpha-104. And then finally, these slides show MS results confirming for 2 of these adducts, alpha PVP and acetaminophen, that we indeed do have this formed, and it is where we think it is. All right, so next steps. We need to increase the sensitivity of this approach by at least 1,000-fold, and there are ways of enriching proteins, enriching the preparations for only adducted protein, and we expect a large increase in sensitivity that way. We need to ultimately make standards, which we're in the process of doing. But finally, the bottom point is this needs to be transferred to an LC triple quad approach so that forensic labs can use this in a more routine manner. So with 10 seconds left, I'll finish with my acknowledgments. Obviously, we thank NIJ and the students and postdocs and staff in my lab who have assisted with this. Thank you.
All right, thank you so much, Dr. DiCaprio. We are going to move to our next presentation. Our next presenter of the day is Ms. Amanda Moore, who will be presenting on retrospective identification of synthetic cannabinoids using archived high-resolution mass spectrometry data. Ms. Moore is a graduate of the University of Montana, graduating with dual degrees in human biological sciences and sociology with an emphasis in criminology. She then obtained a Master's of Science degree in forensic science from Arcadia University. She currently serves as a forensic scientist at the Center for Forensic Science Research and Education, where she works on a variety of analytical projects associated with toxicology samples, teaches graduate students on analytical and instrumental toxicology lab, and serves as lead investigator on several research projects funded through the National Institute of Justice and National Safety Council. All right, we're going to switch everyone's layout now to Ms. Moore's presentation. And without further ado, I'd like to hand the floor now over to Mandy Moore.
All right, good afternoon, everyone, and thank you for joining us this afternoon, and thank you, Josh, for that very nice introduction. So before I begin, I have a quick disclosure. This project was funded by the National Institute of Justice, so the opinions, findings, and conclusions are those of myself and my co-authors. So as I'm sure most of you are very well aware, since 2008, there has been a steady influx of synthetic cannabinoids on the recreational drug market. 181 synthetic cannabinoids have been reported to the EMCDDA in Europe as of January 2019. When we look at what's going on here in the U.S., according to the 2017 NIPOLIS report, synthetic cannabinoids accounted for about 2% of all the drug reports, with over 24,000 total reports. When we look at some of the most recent data available from the DEA Emerging Threat Report, there was 526 synthetic cannabinoid identifications in 2018 in the United States. And shown up there is data from their Q3 report, which shows some of the most common compounds that included 5-fluoro-MDMB pica and ADB-fubinaca. Recently, synthetic cannabinoids have been increasingly implicated in several emergency room admissions, death investigations, and high-profile intoxication events within correction populations. So understanding the scale and scope of this problem requires comprehensive analytical testing. And from a laboratory standpoint, the challenges of remaining current with this synthetic cannabinoid epidemic stem from the diversity of compounds in the class, the large number of analogs and configurations and isomers, delays in the availability of analytical standards, and a laboratory's ability to develop and validate analytical methods quickly to confirm the presence of these compounds in biological samples. So based on these problems, the objective of our research was to develop an optimized synthetic cannabinoid method using high-resolution mass spectrometry where biological samples and extracts could be continually analyzed, creating a historical data archive that could be reintegrated as new compounds emerged. So this would allow us to determine retrospectively the date of first appearance and spread of new compounds as they're identified on the market. And it would allow us for us as well to comprehensively test for synthetic cannabinoids that may have not been incorporated into the scope at the time of testing. And using this approach would allow us to kind of increase the rate at which compounds not previously characterized are identified and reported, and hopefully decrease the lag time between spikes in popularity and the time of first detection, and ultimately allow us to kind of create some time course positivity trend reports, geographic heat maps, and provide basic demographic information related to these compounds. So our method was designed to analyze for both blood and urine specimens. Blood samples was extracted using a basic liquid-liquid extraction with tris HCl into MTBE. Urine samples were initially hydrolyzed and then extracted using a solid phase extraction with the adjuvant blonde elute plexa PAX SPE columns. And then, excuse me. They were, all samples were dried to completion and reconstituted into mobile phase. Chromatographic separation was achieved using a Shimadzu Nexera UPLC using 10 millimolar ammonium formate and methanol acetonitrile 50-50 on a Phenomenex C18 column. Our initial method was a standard reverse phase gradient that had a 15-minute runtime. However, we spent a lot of time optimizing that so we could increase the efficiency and throughput for the samples. So we ended up with a final of 7-minute runtime, and the gradient for that runtime is shown in the column on the right hand of the screen. The other thing to note is that this method was being designed for the separation of known and unknown synthetic cannabinoids. We didn't necessarily seek to separate isobaric species, but we wanted to ensure that all compounds were eluting within the runtime. And we decided at that time that if there was separation needed for isomeric compounds, additional techniques would be sought at that time. With respect to the MS parameters, it included using a scan range of 100 to 550 Daltons on a SciEx triple top 5600 plus. We had a total of 27 swath acquisition windows with 25 Dalton windows near the upper and lower end of the mass range and 10 Dalton windows near the center. All analytes were analyzed in positive electrospray, and there was a collision energy spread between 35 plus or minus 15 electron volts. And for the fragment mass acquisition, we set that to 50 to 550 Daltons. In our initial method, we had 234 parent synthetic cannabinoids and 22 metabolites, along with 16 internal standards. Once we developed and optimized that runtime, we did a fit-for-purpose method validation, performance characteristics evaluated, including the precision and accuracy, limits of detection, interference, process sample stability, and carryover. And we evaluated this for 19 synthetic cannabinoid parent compounds and 19 metabolites that sort of spanned a range of the generation and chemistries, so we could ensure that the method was suitable for all of these different chemistries of synthetic cannabinoids. Excuse me. To determine the limit of detection, For each of these, we did serial dilutions. And then we also, our evaluation criteria included ppm error, retention time error, isotope difference, library score, and peak area. And we expected less than a 20% CV for all criteria over the course of 3 days. Shown here are the 19 synthetic parent drugs and metabolites that were included in the evaluation. So you can see, again, we have some of the first generation synthetic cannabinoids like JWH018, to some of the more recent synthetic cannabinoids like 5-fluoro-ADB, and then a variety of metabolites as well. At the conclusion of validation, all parameters were met for both the parent compounds and the metabolites in urine. There were no interferences from more than the 250 therapeutic abused and emerging drugs evaluated. There was no matrix interferences, and the method was free from carryover. The limits of detection range from 0.05 to 1 nanogram per milliliter in blood, and for urine it ranged from 0.05 to 5 nanograms per milliliter in urine. So following our validation, the next phase of the project was to obtain sample extracts which were provided by NMS Labs and re-inject them on our optimized method. And then all of the samples were processed against our library, which I mentioned contains over 250 parent compounds and metabolites. And then subsequently, we used that data to develop real-time quarterly reports, provide some trend data and demographics. So shown here is the demographic data acquired between October 2018 and March 2018. And if you'll follow along with me, you can see here, beginning in November, we start to see a spike in positivity for 5-fluoro-MDMB pica, which kind of peaks again in January and starts to trail off. We can also see in November we have some increase in positivity for 5-fluoro-ADB, which again subsequently falls off after about December. We also see beginning kind of in December this uptick in positivity for 5-fluoro-MDMB pica, which I just mentioned, and then their metabolites, their corresponding metabolites, peak in positivity and follow the parents. It's interesting to note this was for blood and urine extracts, so we were detecting metabolites in some of the blood extracts, which was something that was previously reported at one of the meetings for forensic toxicologists, but this is a trend that we're seeing, that you are able to detect the synthetic cannabinoid metabolites in blood as well. For our positivity data, this is looking specifically at Q1 of 2019. There was about a 12.8% positivity for the parents and metabolites. During that time, we analyzed 1,533 extracts. Our highest percent positivity was for 5-fluoro-MDMB pica, followed by 4-fluoro-MDMB binaca. And this was kind of the first time that we had seen these two detected in the highest frequency. Prior to that, the highest positivity rate was for MMB flubinaca and 5-fluoro-ADB. So this is the first time that we've kind of seen a turnover in terms of positivity with those two dominating the positivity in our samples. The new thing with respect to synthetic cannabinoids seems to be some increase in combinations. So what we're seeing in terms of combinations is 4-fluoro-MDMB binaca and 5-fluoro-MDMB pica. Together, that was positive in 9 samples, so not a huge percent positivity, but again, it's there. Those two structures are shown at the bottom of the screen. And then we also had a few samples that had, in addition to that, some 5-fluoro-ADB. Here is kind of the demographic data associated with the positive cases. So you can see about half of them are turning up in postmortem cases, about a third in DUID. They're primarily being detected positivity we're getting out of blood samples. And the vast majority of these positive cases were males and a company in a range of 21 to 50. Shown here is the quarterly heat map of positivity. So you can see we have a hot spot in Pennsylvania and some positivity in Texas as well. Again, the limitation of this is limited by the fact that not all agencies are submitting their samples to NMS labs for testing, so it's not necessarily representative of all jurisdictions or a true positivity rate of synthetic cannabinoids in the United States. One of the other populations we are very interested in studying was correction populations because here in August, the Pennsylvania Department of Corrections went on lockdown due to prison officials being sickened, and that was suspected to be due to synthetic cannabinoids, and they had a large influx of synthetic cannabinoids within the prisons. So we are currently receiving urine samples, de-identified urine samples from 2 sites. Out of the 176 samples that we have tested so far, there have been 9 positives. 6 were positive for 5-fluoro-MD-MB-pica metabolite only. 2 were positive for 5-fluoro-MD-MB-pica, that metabolite, as well as the 4-fluoro-MD-MB-binaca metabolite, and 1 was positive for the 4-fluoro-MD-MB-binaca metabolite only. As you saw from the title of this presentation, one of the large goals of the project was to be able to retrospectively go back and data mine using this archive that we had created. So our library database is continually updated on a monthly or weekly basis with new synthetic cannabinoids as we either hear about them or they become available through standard reference material vendors. And this allows us to look for these new compounds in samples without the need for re-extraction or retesting. And we primarily obtain our information from seized drug material, drug user forums, scientific meetings and network, and then we frequently visit the standard reference material vendors' websites to see what new materials they have out there. Since validation, 12 compounds have been added to the library. The list of compounds is shown in the table at the bottom of the screen. So this includes some of the new compounds like chemopegaclone and 5-fluorochemopegaclone, which we heard was getting some positive hits over in Germany, 4-fluoro-MD-MB-binaca and APP-binaca. I believe the 4-fluoro-MD-MB-binaca was initially alerted to us by some of our colleagues in Singapore. So once these were added to the library, all data was subsequently mined. So with the addition of 5-fluoro-MD-MB-binaca, we have identified that that drug in 34 cases since December of 2018. The data, the first sample collection, dates back to late November, and it was initially identified in a post-mortem case. This drug has been identified from 10 states that include Arkansas, Connecticut, Florida, Indiana, Kansas, Michigan, New York, Pennsylvania, Texas, and Utah. And currently, 4-fluoro-MD-MB-binaca is not a scheduled drug in the United States, and it wasn't part of the original scope of testing as well. This is just showing the data associated with that initial positive. You can see it had a retention time of 4.48 up here. Sorry. 4.48, and then down here we have the spectrum relative to the library spectrum, and you can see those match pretty well. The other analyte that we have detected for the first time in our analysis performed in Q1 was APP Banaka. We had 7 positive cases since December of 2018, with the first date of sample collection dating back to early December, again, initially identified in a postmortem case. This has been identified in cases from Pennsylvania, Indiana, Louisiana, and Texas. Again, and it's not currently a scheduled drug here in the U.S. And then, again, there is just the data from that first positive hit. So just to kind of bring this all together, we have developed an optimized and analytical method for the detection of synthetic cannabinoids, and we have created a high throughput method that originally started out as a 15-minute, and we were able to decrease that run time down to about 7 minutes, excuse me. Between January and March of 2019, we analyzed 1,533 extracts and had a positivity rate of around 12.8% for at least one synthetic cannabinoid parent or metabolite. These new synthetic cannabinoids still continue to persist on the market, with a new one appearing about on a quarterly basis. And what we've seen from our trend data is that peak positivity is generally within 3 to 6 months of the first detection. So again, this really creates a challenge for laboratories to keep up with the pace of this evolving drug market. But what we have seen is that there is a high degree of utility in developing a historical data archive, which allows you to identify when new synthetic cannabinoids first appeared on the market. Again, while our geographical distribution data set is limited, we have some indication of the spread of these compounds and where they are, and we can watch rates of use and trends over time by continually mining compounds. New synthetic cannabinoids are going to continue to emerge, so this seems to be an effective way to identify them in cases. And without real-time sample mining and reporting, we're always going to be behind the newest trends. And this has some pretty significant implications for casework. As I said, those 2 new compounds that we've just recently identified weren't initially part of the scope of testing, so if that sample was submitted only for synthetic cannabinoid testing, they could have went out as negative results. If you are interested in our identifications or our quarterly reports, you can visit npsdiscovery.org. That's where all of the information is uploaded on the website and accessible to you. And I think there's also an option on there to subscribe to the list, so you'll get notification emails when new discoveries are made or when trend reports become available. We have a few acknowledgments. Again, there's multiple people that play a role in this, so I would be remiss if I didn't acknowledge the following people. And with that, I would be happy to take any questions if you have any.
All right, thank you so much, Ms. Moore. To keep everything on time, we are going to move on to our next presenter of the day. Our third presenter in today's event is Dr. Carl Wolf, who will be presenting on the development of matrix-matched quality control materials and sample preparation techniques for the analysis of marijuana-infused products. Dr. Wolf received his Bachelor's of Science in Chemistry from Gannon University in Pennsylvania in 1986, where he received the CRC Press Outstanding Freshman Chemist Award. He then received his master's degree in criminal justice with a forensic science option from Virginia Commonwealth University in 1994, and then achieved his PhD in pathology with a focus on forensic toxicology from the Medical College of Virginia campus at Virginia Commonwealth University in 2005. And he has been employed at the Medical College of Virginia Hospital since 1987 in various roles in the clinical and forensic toxicology laboratories. And we're going to switch everyone's layout now. And I'd like to hand the microphone over to Dr. Carl Wolf.
Hello, everyone. Welcome. We'll get started here, and as always, put my disclosure statements. We want to thank all of you for paying your taxes and for NIJ funding us. These all opinions in this are mine and not those of the hospital here or the university or any of its employees, and we're going to be talking about a number of commercial products and other things, but there is no endorsement. Let's see if we can start. So this whole project actually started out in April of 2016. Reading one of the many 80-some emails that I get a day, this popped into my inbox and said, hey, there's new pot business going on in Ketchikan, Alaska, and I was taking a trip there that summer, so I thought, let's follow up on this and see what's going on. State law requires that THC content be tested, but as they said, there are no real testing facilities there on the island to do this. So what are they doing? And it sort of popped into my head right away going, now we have an issue because even if they do have a lab there, where are you getting your matrix mass calibrator and control materials? A very relevant question, especially for myself being in the hospital setting here for the last 30 plus years is, we don't do urine testing off a blood curve and vice versa. So, and many of the accrediting agencies require some kind of matrix match calibrators and controls before you can go ahead and do any work because, as we all know, matrix is a big issue. And why would there not be any other difference going on? So on my trip to Alaska, I like to throw this in here, I asked one of the vendors, jewelry vendors when I was in Juneau and said, hey, where can I find a dispensary? And he looked at me kind of strange. And I explained to him who I was and what I was trying to do, looking at cannabinoids in food products. And he said, oh, come back in 2 weeks when they figure out what they're doing. And that seems to be at some point in time along the last 3 years sort of the statement that I've gotten from a number of people I've talked to about this is how is it being handled? So we went forward and said, okay, what do we need as a serving? And states have defined servings as either 5 or 10 milligrams of THC, but no one has really put in what a serving of CBD is. And CBD, up until recently, or at least over a year ago, has been in the news about all of its therapeutic effects and helping people and such. So we thought, we should include that too in our testing that we're looking at. We thought, what's the best way to start? Pot brownies. They've been around for a long time. What's a serving of a brownie? Duncan Hines and many of the other manufacturers say that there's about 20 brownies in an average box, and there's about 50 brownie bites per box. So we said, cool. We have 2.5 brownie bites as a brownie, and we have a serving for each. So let's make some control materials with 5 and 10 milligrams of THC and CBD. And this is basically how we put 2 milligrams in each bite or 4 milligrams in each bite, and that became our standard that we were working with. We got out our personal protective equipment. We decided that cake brownies are appropriate because when we make chocolate brownie trifle for Christmas, the cake works much better. But we made it according to the box. Everything was prepared according to the box. We heated our oven up 300 degrees per high times recommendation because cannabinoids start breaking down above 300 degrees Fahrenheit. Put it all together. We divided our batter to make sure that we had the correct proportion afterwards. We fortified it with the QC. We put it in our brownie tins. We were and we baked it at 300 degrees in our brownie bite pan. Pulled them out, made sure that they were done with a toothpick because it took a little bit longer in your conventional laboratory oven versus your home oven, somewhere around about a half an hour time. Took them out, cooled them, and we moved forward. We had both our blank matrix and our 2 control materials we made. We labeled them appropriately so no one would even think of trying to eat them here. We then borrowed the Washington State Crime Lab's method for doing baked goods. It's a thermo application note. Seemed to be fairly simple, straightforward, 25 milligrams of sample, 2 mils of methanol, mix them well, equilibrate, filter, and then do a 1 to 50 dilution before you put them on the instrument because you're running them in such high concentrations. We did some precision studies, some bias and process efficiency, and we were looking at we can do fairly well at extracting both THC, CBD, and THC deuterated out of the samples, and we got good bias and CVs on our triplicates that we were running. So we said, okay, let's run this for a year and see what happens. And as we see, these were actually sitting in my office for 1 year in plastic bags. And over the 1 year time period, we do get some variability in results. Some of that we think sort of in the 5th and 6th month may be due to more instrumentation than actual samples. But at the end of 12 months, other than the THC in our 5 milligram sample being a little bit on the low side, everything else seemed to be fairly consistent. We said, okay, brownies are great, but there are a ton of other matrices out there and marijuana products. And we are working on a couple other projects with some other collaborators doing things like dabs and tinctures. And we were like, well, what's baked goods? Let's look at the food product side of this thing. You know, what do we have? We have candies, we have drinks, and we said, what are the analytical challenges we run into doing these type of things? One is the cannabinoids, and not just THC and CBD, but the other cannabinoids, their concentrations vary greatly based upon the plant variety, the growing conditions, the geographical region for these. In our medible matrices, as we called it, our marijuana edible matrices, we said, well, you have 3 basic things you're looking at: high fiber, high sugar, and high fat content foods. We want to look at each one of those individually, not necessarily together. There's homogeneity issues with any batch, and if someone were to send you the samples, or if you were to sample from your batch that you make, is it truly representative of the whole thing? If you're making products, do you do the sampling to the materials pre or post formulation? There are no federal guidelines or standard methods out there really for doing testing in these products. There have been a number published, but we have not really seen any validation data. Interstate transportation of cannabinoids is still unlawful. And one of the questions was storage conditions. So if you're in the lab, do you know what happened to the sample before it got to you? Do you know what happened to the sample once you receive it in the lab? Where do you store it? Do you store it in the back of the police car for a month in the middle of summer? Do you put it in your room temperature, which varies? Do you refrigerate them? Do you freeze them? Or do you have to drop them in in the deep freeze in the -70 freezers? The other thing is getting enough reference material, as we found in our first study, to just try to make up a large enough size batch to use for QC materials so we could test them over the year. That created its own issues of being able to only buy about 1 milligram of sample at a time, DEA exempt, and finding a source for higher ones is not really that easy. So we were looking at our medibles, our brownies, gummies, and chocolate, most importantly chocolate. We went back to the analytical challenges and we said, hey, what can we do here to make this thing work? What of these can we look at? And we said, well, we can definitely look at edible matrices. We can test for those. We can come up with a standard method that works across all 3 matrices. As we've done some work, and we'll see this in here, each matrix is unique to itself, and even though they're food, as we would know from doing human body testing, blood is not the same as urine, it's the same as liver. So, and then what storage conditions, because there are nothing really published out there other than our study that we did a few years ago. So we want to look at food first. Edible matrices, what's out there? Well, high fiber, we picked brownies. Candies, we decided to go with gummies. They're a little easier to work with than hard candy, and they're a little easier to make. And then, of course, the universal food, chocolate. Dark chocolate, because as reviewing many online websites for available products, dark chocolate seemed to be the preferred matrix, probably more for the fact that the terpenoids in marijuana have a little bit of a bitter taste, so if the chocolate's a little more bitter, you won't get as much issue with that. So we already had our defined definition of a serving of brownies. Gummies, we went to the Haribo because they seem to be the most commonly out there, and 39 grams is considered a serving, or about 17 bears. And for chocolate, we went to Hershey's, we went to Nestle's, we looked around, but we went for the standard 41 grams is 9 Hershey's Kisses or 4 Hershey's Nuggets. So we made our brownies as we had done before. Not a problem. We wanted to evaluate fudge brownies versus cake brownies because there's two out there and which one works better for our project. Gummies, we had a student who presented this back in 2016 or presented at the Academy in 2017, but spent 6 weeks figuring out how to make gummies. Basically a box of gelatin, some unflavored gelatin to get it to set up better, a little bit of water, and some mixing and such and putting it together. Chocolate, well, what is chocolate? We have dark chocolate definition, we have milk chocolate, we have white chocolate, and not that nasty white stuff that somebody tries to call chocolate, but there's an actual definition for white chocolate. And then we had to do, well, we're making a product. Is this even a real product? Would anyone ever consume this potentially if this was to be turned into a medicine? And so we looked at if we hand make ours versus a refiner or a semi-automated approach. So after doing that, I should say we had a resident here who took on this project and performed palatability testing. We looked at 5 different recipes and tested them blindly on individuals who were more than willing to assist in our project. No, there were no cannabinoids in any of these. It was just blank matrix. We then went forward and found out that there was 2 sides to every coin. We had the milk chocolate lovers who hated the dark chocolate bitter taste, and the dark chocolate lovers who hated the simple taste. This was our formula that everyone seemed to enjoy. It's about a 65% cacao, and we make this by hand in the lab in my office. It's 60 grams of cacao, 30 grams of cocoa powder, a little bit of honey, and a little bit of vanilla and oil. With the directions there, takes about 10 minutes to 15 minutes to make this up. We let it cool to room temperature. The big issue with chocolate is homogeneity, because if you don't line up the crystals correctly, we get fat blooms. And as we know, THC and the cannabinoids are fairly lipid soluble, so we're going to lose our consistency through our product. It's also what we see sometimes commonly in products that we buy from people. If they're not tempered correctly, we don't get that nice shiny gloss that we like to see, and we don't get that nice crisp snap that we expect to see. Tempering involves lining up basically the fat crystals and all the crystals in the chocolate together in one direction. So you take out about 1/3 of your chocolate, you chop it up nicely, you heat your dark chocolate up to 114 to 118 degrees Fahrenheit using a thermometer. We slowly add back in the seed in divided portions. We take it off the heat. We stir it until it reaches about 85 degrees Fahrenheit. We take a portion out, add our cannabinoids, and then mold it up in our lovely coffee bean form for simple storing, correct size. The other thing was analytical methods. So what are we doing? How are we making this work? We're looking at 9 cannabinoids in doing this, and some carboxylic acids. It's quite a different variety of compounds to look at. We had a method that was published as our candy method, as we call it, back in 2010, where we were able to separate all these, and it uses a methanol water with a 0.1 millimolar ammonium formate solution, and we can easily separate all compounds of interest. Those with the stars, we looked at 3 deuterated internal standards, calibrators, and control materials that were prepared in our matrices. We looked at one thing we learned from doing a previous NIJ study was looking at matrix effect, recovery, and process efficiency, because if the method doesn't work for those 3, it's not worthwhile going forward doing anything else. And we like to have our matrix effect within plus or minus 25%, and our recovery 75 to 125%, as well as process efficiency. Won't put the formulas in there because people should easily be able to calculate that. So we looked at just a few things here with our students and myself. We looked at dissolution. Can we just dissolve these compounds in common commercial solvents we have available in the lab, or organic solvents we have? We even tried water. We looked at liquid extraction techniques. We looked at filtration techniques and whether or not we buffered them, and the manufacturers there. We looked at solid phase extraction techniques going, can we get this to work some other way? And then we looked at hybrid techniques, with and without adjusting the pH of the sample, basically using a catcher's method from 2 different manufacturers. So results of the basic thing are between the 3 matrices we're looking at, fiber, sugar, and fats, trying to just dissolve these samples in hexane or something else, or extracting them in hexane or any one of the other number of ethyl acetates seemed to be promising at the moment. We thought methylene chloride was wonderful until we found it actually destroys your cannabinoids. Whether you extract them or whether you just add them to methylene chloride, within about 5 minutes, all the cannabinoids were destroyed. So we were like, this solution is not going to work. None of those. Liquid-liquid extraction. Again, across all 3 matrices, we saw no consistency whatsoever, no matter whose manufacturer we looked at. We then went on and said, well, these are fat compounds. Manufacturers make filtration products and some other things that we looked at. Again, no consistency across our matrix process efficiency or recovery. We then went on to our solid phase extraction techniques. In those, again, we saw nothing that worked for all 3 matrices, because the problem with this is it's not so much, oh, can we run a brownie, can we run a gummy, or can we run chocolate, is what do you do with the chocolate chip cookie or the chocolate chip brownie with gummy worms in it, the dirt sundae approach. If you've got a sample like that to analyze, you can't pick it apart, you have to run it for full content. But we ran them separately just to see which would work the best or not work the best for each method. Catcher's method, we went forward with that. This is the first one where we actually saw some reasonable expectations of acceptability. Red is bad, green is good. But we see there are some problems here with our things. We're getting some recoveries that are above 100%. We're getting some process efficiencies that are above 100%. Not bad from all the methods we tested, and we were going to go forward with this. We then got introduced from Phenomenix, who said, we make a catchers too. So we went to evaluate theirs. They request that you add a 1% acetic acid. As you see, red is bad. Didn't look too well. We were not happy with it. But we had learned through another process of doing some earlier studies that we don't need to add the acetic acid. We dropped the acetic acid from it, and our results look a whole lot more consistent across all matrices and across at least our compounds of interest, which are really THC and CBD, and their deuterated internal standards. So this is the method we went forward with. A simple breakdown of the method, 25 milligrams of sample. We add our cannabinoid mix. We add 1.5 ml of methanol, 1.5 ml of acetonitrile. We let them sit for an hour. We add your internal standard. We vortex them well. We add 0.65 grams of the rock salt, that's the roque salt. We shake them for 30 seconds in a bead ruptor at 5 meters per second speed, because mixing is very important at this step. We centrifuge them. We transfer the acetonitrile into the DSPA tube for solid phase extraction, vortex them, and then we transfer the supernatant to a glass insert auto-sampler plate, because as we found out, if we run them in straight up plastic, they're gone in literally 1 day. So storage conditions, as we're running out of time here. Basically, we stored them, we made them in various matrices. We stored them either vacuum packed or zipper packed. We stored them at room temperature, refrigerator, freezer, and deep freeze. As we saw, we had problems with them in various things, but these are how we made each sample up. Stability, fudge brownies worked much better than cake brownies. Gummies without preservatives lasted longer. Dark chocolate did nothing. We needed to vacuum pack all the bags to make them work well, and that work is continuing on at this moment for a future presentation. Conclusion, there's many different types of matrices, and each is unique, and specimen storage matrix also makes a difference. Handmade chocolate is preferred, and the biggest problem we had was obtaining enough reference material to do what we were doing. The modified rote method worked the best out of what we saw in all three matrices, and we found out that mixing the samples extremely rigorously is very important. Thank all the folks, including National Institute of Justice, my co-collaborator Justin Poklis, the Liver Girls who worked on the beginning of this product, Katie Orton, who has worked since then, Dr. Fielden, who did all of our palatability testing, and of course, Virginia Commonwealth University and the lab here. Thank you.
All right, thank you so much, Dr. Wolf. We're going to switch to our final presenter of the day. Our last presenter is Dr. Chris Mulligan. He will be presenting on coupling Raman spectroscopy with ambient sampling, portable mass spectrometry for on-site high-throughput evidence confirmation. Dr. Mulligan is an associate professor of analytical chemistry at Illinois State University with over 15 years of experience in developing miniaturized analytical devices with specific expertise in portable and miniaturized mass spectrometric instrumentation development, novel ionization methods, and application development. Through his research, Professor Mulligan seeks to demonstrate the impact and practicality of portable mass spec systems for use in crime scene investigations, law enforcement, and security applications. Without further ado, we'll switch everyone's layout now, and I would like to hand the microphone to our final presenter of the day, Dr. Chris Mulligan.
Well, thanks much for the introduction and the opportunity to discuss some of our NIJ-funded work with portable instruments for forensic drug analysis. So I'd like to begin by briefly discussing our motivation for this work, for which a recent crime lab census from the Bureau of Justice Statistics is quite helpful. So looking at the 2014 data, it becomes apparent that controlled substance requests play a major role in both the typical lab workload and also this ever-present sample backlog. Controlled substance requests were about second only to forensic biology, taking up about 33% of all the requests, and at the end of 2014, it accounted for about 37% of the national backlog. As expected, these controlled substance requests accounted for the highest workload for county and municipal labs, and this shows the burden it causes at the local level. So these statistics illustrate the sheer magnitude of the drug evidence workload, and we propose a twofold solution to alleviating this burden, and that's to increase the throughput of drug evidence processing while also decreasing the amount of evidence required to be sent to that offsite lab. And what this gets you is that it allows public labs to divert more of their resources towards other rigorous evidence types, such as DNA analysis, and it could ultimately lead to a reduction in backlogs and the need for outsourcing. Now, when considering evidence analysis, methods that allow direct sample investigation are appealing due to their rapidity, but the highest throughput would come from the ability to actually do these analyses in the field. So this pursuit of throughput led us to merge two different areas of mass spectrometry, that being ambient ionization methods, which allow rapid screening of samples in their native state, and also portable, rugged instruments for field operation. Utilizing a suite of robust, operator-friendly ionization methods, we set out to demonstrate the ability to broadly screen forensic chemicals of interest, and doing this from common evidence types with these portable systems through our past NIJ grant work. Such a system, in turn, we feel would allow only the most pertinent samples to be sent off to the traditional crime lab for secondary confirmation, and it offers benefits such as reduced burden of this drug evidence on the backlog, potentially expediting investigations, and potentially serving as a shareable asset. Now, today, in terms of our discussion, we're going to focus mostly on adapting these methods that I talked about here in terms of field confirmation of evidence, with the target outcome of only involving the crime lab when absolutely necessary. More specifically, what we're doing here is combining a technique called paper spray ionization, which employs paper substrates for an electrospray-like ionization method, with unique metal nanoparticles that allow us to do surface-enhanced Raman spectroscopy, or SIRS, as I'll refer to in the talk, from a single paper swab. Now, What we hope that gets you then at the end of the day is the capability of looking at bulk and trace drug evidence with two independent, highly discerning techniques, which we hope meets the requirements set forth by gatekeepers like Swig Drug in producing this core admissible evidence in the field. So I'd like to point out also that this work is being completed by an interdisciplinary team of investigators at Illinois State University. Mistake. So for these studies, we chose a commercially available MS testbed, this FLIR Systems AIMS 1.2. And a couple things to note here is that it offers MS capability, which allows us to use fragmentation data to do chemical identification. More importantly, though, it features a continuous atmospheric inlet, which allows us to sample ions generated from said ambient ionization techniques. While it's not hand portable, it is amenable in terms of size and weight, and you can transport this directly to a crime scene or operate it from the back of a law enforcement vehicle. It's important to note that the instrument itself provides everything necessary for MS and MS-MS analysis. The only thing you need to supply is power, which is very flexible in terms of the inputs, as you see here, with 110, 220, and 24 volt. Now we've modified this instrument to include this mounting system that allows us to incorporate modular ambient ionization methods, as you've seen right here, in a plug-and-play type style. So a significant aspect of our past NIJ funding was demonstrating these ambient ionization methods for ruggedized portable instruments and showing the applicability overall. So what you see here is 5 different ion source and evidence type combinations in less than 6 minutes. So the ion chromatogram you see at the top shows data collected for evidence in real time, with the baseline in between, with that green star that you see right there, corresponding to the time required to swap out the plug-and-play sources. Here we've screened Coleman fuel vapors, which is of interest to clandestine drug manufacturing, using a modified atmospheric pressure chemical ionization source. We've looked at MDMA residues from solid surfaces with desorption electrospray ionization. We looked at a residue of phenethylamine derivatives swabbed via paper with this paper spray ionization technique. Direct tablet analysis with paper cone spray ionization, which is kind of like a 3-dimensional paper spray. And then lastly, a more traditional extract of a drug sample in methanol via electrospray ionization. So overall, such a strategy allows the operator to select the optimal technique for the evidence in question while minimizing the overall sample preparation at hand. Now through these efforts, paper spray ionization kind of stood out as having the broadest applicability and also the easiest to master in terms of novice users. So paper spray utilizes a triangular piece of paper, this paper substrate, as both the sampling medium and a disposable ion source. So after the said paper is used to sample a piece of drug evidence, either through dipping, spotting, or surface swabbing, we add a compatible solvent and we add some high voltage to produce an ESI-like ionization method directly from the tip of the triangle. So this turns into your ion source in that way. Also, deposited analytes are then released from the paper after they are solvated, and you collect those ions via the MS inlet, as you see in this photo right here. Now, also seen here is how it fits into our modular design, and then lastly, you see how we can use said paper swabs to look at everyday evidence, such as latent fingerprints on door handles. But let's see how it actually performs with authentic drugs. So some of the data is presented here is through collaborations with local police agencies. In the top left-hand corner, we're looking at direct analysis of bath salt compounds. You see here this is a DISCO all-purpose solution seized locally at a truck stop. Actually analyzing that white powder substance, we see that it contains penadrone. The bottom left-hand corner is some of our work that we've seen with alpha PVP, not only in terms of high-quality illicit tablets, but more importantly in terms of adulterated e-cig liquids. So looking directly at that adulterated e-cig liquid, what you see is signatures not only for alpha PVP, but also nicotine and methanol, as you'd expect from that matrix. Top right-hand corner is looking at blotter paper with N-bombs, or these 2,C-phenethylene species. So that's looking directly at the paper cut into a triangle, as you see. And then lastly, injectable drugs, particularly the fentanyls, opioids that we know is of major interest in terms of illicit drugs nowadays. Now the capability of these techniques also allows us to look at evidence that might be typically overlooked in a traditional investigation, such as paraphernalia. So we see here presented looking at a spoon used to inject heroin, producing the signals that you'd expect. And on the right-hand side, this is actually a field-tested sample that field-tested positive for cocaine. When we actually investigated it with the paper spray technique, we saw that that was actually a crushed-up trazodone tablet, even though it did test positive in the field. So it shows the capability of providing these accurate identifications. Now, obviously, since it's going to be used by non-technical users, it's of increased interest to make sure that it's going to operate reproducibly every time that we use it. So what we set out here to do is to discern the overall error rate from different types of user classes. So we set out, we trained everyone for roughly 50 minutes, and then we set them loose looking at positive and negative controls. We see that indicated here by the detection rate and the false positive rate. And overall, what you see is that the outcomes scaled with the education levels overall. But overall, as the more novice users, like high school users, the more that they utilized it, the more the results started to match that of the most trained users. Now, also notable here is at the end of the day, when we look at our detection rate and the false positive rate, that it starts to approach that very recently reported for DEA-affiliated labs. Also going back and look at some of the false positive and false negatives that were measured, we were able to attribute most of those errors to user error. And that shows that through some extra training, a little bit extra experience on that, we hope to rectify that and make these rates even better. Keeping in mind with validation, we also want to make sure that it can operate in the field. So a lot of our efforts went to show potential environmental effects. So what we see right here is a heat map that looks at signal intensity with paper spray across varying outdoor temperatures and relative humidities. And this can serve as a guide to inform operators in terms of the anticipated sensitivity of the technique. Now overall, it's important to note while there are some red zones here which correspond to low intensity, if you will, never did we actually experience errors because of that. This is only set to kind of guide overall performance. Mostly this red zone corresponded to a twofold type reason. That's a rapid evaporation of the solvent from the paper and also user duress. And I'm sure my research students who collected this data at 95 degrees outside can attest to that. Now that we've established the potential of paper spray by itself for field investigation, let's discuss the strategy of incorporating an independent Raman analysis from a single sampling swab. So here's our proof of principle rig, if you will, to perform this technique. You see from this little breakout here where we're positioning the Raman laser probe, and we also see that the paper triangle has been functionalized with nanoparticles. And this is what allows us to do a SIRS analysis. That SIRS analysis overall allows us to do very trace molecular detection. But since Raman is non-destructive, we're also able to conduct the paper spray technique from that single swab. A primary goal of this initial, or I should say a primary goal during the initial phase of the work was to establish whether we could find a metal nanoparticle system that could be applied directly to the paper substrate that allowed us to not only do sensitive SIRS analysis, but make sure that it's compatible for paper spray as well in terms of any potential chemical background or interference with the spray characteristics of our device. Now, some of the candidates investigated are seen here, such as spherical gold, spherical silver, gold-silver core shell, and anisotropic gold designs. Also shown here are absorption and scattering profiles at the 785 nanometer wavelength of our Raman probe. And once a suitable candidate for both SIRS and PSI was established, we set out to characterize this combined technique. And with that, I'm pleased to present some of our most initial data here in terms of actual authentic evidence analyzed. So what we really sought to show with this fieldable technique is a way to maximize the strengths and minimize the weaknesses of each of the individual methods, that being paper spray and this SIRS technique. For instance, it's well known that mass spectrometry is quite adept at mixture analysis. And here we have a mixture containing various phenethylamines and MBOME derivatives. More specifically in here, we have a 2CD, which is a methyl moiety, 2CN, which is the nitro moiety, and 25I-MBOME, which corresponds to iodine in that X position you see in the molecules provided. Looking at the base scan data, we see several prominent peaks, but given the MS-MS capabilities, we're able to go through and sift through these and determine whether those correspond to a protonated molecular peak of a drug of interest, potential in-source fragments or adducts, or peaks stemming from street drug matrices such as cutting agents. So this can also be done with onboard software. We don't have to do this necessarily visually. We have onboard software that we've modified to also perform automated database matching and provide a color-coded response whether there's an alarm for a known drug that's in our database of interest. But it's also well known that MS can be susceptible to isomeric compounds, even in the forensic lab with GCMS systems. Here we are presented with 3 isomers of a synthetic cannabinoid, and this is of the JWH-018 class. So all 3 compounds here show the same protonated molecular peak at 342. I'll kind of highlight this, so 342. Each one of these compounds of interest will give said base scan data. If we look at the subsequent MS-MS analysis of each one of these techniques, and you notice that these are color-coded, you see that they're nearly identical in terms of the MS-MS spectra that you get. So effectively, from a mass spectrometry standpoint, these produce the same exact data, and we'd only be able to determine that this is potentially JWH-018 or an isomer of it from a paper spray standpoint alone. But now, with the strengths of the SIRS technique that's compatible with the paper spray technique, these in-field investigations stand a stronger chance of isomeric differentiation. So some of our initial SIRS results suggest that through both visual and statistical means, we can elucidate problematic isomers like this JWH-018 system I just showed, as differences can be seen in the specific Raman shifts, particularly as highlighted here in the pink zones at hand. So this allows overall a better chance of an accurate identification of evidence at hand, even in that difficult class of novel psychoactive substances. And again, I point out that this is initial work, but overall this initial work is quite promising. And this finally brings up where we stand today in terms of our efforts, the current work, if you will. And in those efforts, we seek to establish the overall detection limit of each one of these methods and the combined SIRS paper spray technique from different modified paper substrates to, again, find the best substrate that's available to us. As seen here, we have a modest set of test analytes. So we have cocaine, hydrocodone, fentanyl, 2CB of the phenethylamine class, and then we have that JWH-018 species. And we notice that from all the different substrates that are seen here, both paper by itself and also substrates that are modified with the nanoparticle species, as you see right here, that we see that across the board, we see nanogram level detection in terms of detection rate. So we can detect these analytes all the way down to that level. Now you do see some differences between substrates and analytes. Overall, you do see that the SIRS technique is performing a little bit better than the paper spray technique, but as a whole, both of these techniques together allow you to look at that trace analysis capability and also potentially providing you confirmation of your chemical identification. Now you also, I should point out here that with the JWH-018 species, as highlighted here, this is looking at this type of species with 2 different types of spray solvent systems for paper spray, that being methanol or acetonitrile, because there is some solvent compatibility issues, or essentially suitable solvent choice in terms of paper spray. But you also notice that this is not affected in terms of SIRS. The solvent that you choose for paper spray does not affect the overall SIRS analysis. So there are some other instrumental aspects and method characteristics that we're trying to optimize to perfect this technique. And similar to our paper spray efforts, we seek to determine the overall error rate of this technique, and we also need to demonstrate robustness in terms of field operation. So with that, I'd like to thank all the collaborators we have on this project. So like I mentioned, there's multiple groups that are involved in this. In terms of my research group here at Illinois State University, my students seek to elucidate the paper spray aspect of the technique. The Driscoll group has been working on perfecting the Raman analysis, and then the Kim research group at Illinois State University has sought to produce these novel nanomaterials for functionalizing the paper substrates for this combined technique overall. Dr. Jamie Whelan's on the project as well to assist us in the statistical validation of the project, particularly from the error rate standpoint. And it should be noted that all of this work was conducted mainly by undergraduates and MS level researchers because Illinois State University is a primarily undergraduate institution. And lastly, I would be remiss not to thank the continued support of the National Institute of Justice, continued research stream here to provide the funding we need to show the propensity of these portable systems for field drug analysis. And it should be noted, as other presenters noted here today, that the opinions, findings, and conclusions of this presentation are not necessarily reflected of that of the National Institute of Justice. So with that, I'd like to thank everyone for their attention and obviously answer any questions that you might have. Actually this time, because I think I'm last.
All right, thank you, Dr. Mulligan. We're going to switch everyone's layout now. Just to expand the chat pod, we only have just a couple of minutes, so I'm going to pose a question to all of our presenters. We'll start with Dr. DiCaprio first, but it will be the same question for everyone. So just to kind of give us kind of an extended look into your research and where it is heading, as well as when you feel like you might be able to publish the results of your research and your grants.
Okay, so this is Tony DiCaprio. So we've published the one article on the glutathione modifications, which is in Xenobiotica. We have another publication submitted and in review on the synthetic peptide modifications, and we're working on one for the data that we have so far on in vitro hemoglobin modifications. And as I said during the talk, I mean, our main goal here is to try and get this validated and routine so that it can be a viable option that a typical forensic lab, as long as they have an LC triple quad MS, will be able to perform.
All right, thank you, Dr. DiCaprio. Ms. Moore?
Yes, as I mentioned during the presentation, those quarterly trend reports are being currently posted on npsdiscovery.org. Those are available, the last one within the first week of quarter one ending. We had that up, so just about a week into April, we had that Q1 2019 data available, and we plan to continue to have timely quarterly reports available. We also have submitted a manuscript on the 4-fluoro-MDMB Binaca 4-fluoro-MDMB Binaca. That's been submitted to Journal of Forensic Sciences, and that's a comprehensive paper detailing everything from the detection and seize drug work through toxicology as well as metabolism. So that should be coming out. It's in the revision stages now, so that should be out before too long. And then we have a couple other of synthetic cannabinoid papers in the pipeline that should be coming out before the end of the year.
My turn. So we've presented this at a couple meetings, including the Academy of Forensic Science and the Society of Forensic Toxicology. We are currently working on this as one giant manuscript to talk about how to do the testing as its own separate one. That realistically probably will not be finished or submitted until the end of May. And then we have an ongoing where we are, I think, 3 months or 4 months into our one-year stability study in all three matrices at 2 different concentrations, including the carboxy acids, for a 1-year time period. So that in itself is going to be its own separate publication for that. And at the moment, that's kind of where we are with that. And again, we're presenting things as we go along at various different venues and possibilities.
All right, thank you, Dr. Wolf, and
All right, this is
Dr. Mulligan, if you'll lead us out.
Sure. Thank you. Since we're trying to be on the cutting edge here in terms of instrumentation, we really try to take the opportunity to not only present but also publish this as we go. This is a really hot area of research, so if you don't publish it, you're going to eventually read about it because other people are going to be working on it as well. We haven't quite published any of our novel nanoparticle work or our validation efforts on the combined Raman and paper spray technique, because again, we're kind of in the heart of that data collection at the moment in terms of our grant. This summer will be a lot of the error rate investigation, taking it out into the field, determine these environmental effects. Summer is really good for that, as well as practicing the technique on authentic evidence. I should say that much of our paper spray techniques that we've described in this talk, as well as the validation of the paper spray technique, most of that's been published. There's a 2017 article in the Journal of American Society of Mass Spectrometry, as well as other types of shorter communications that talk about application development for the overall instrument. And I'd be happy to provide reprints or anything like that, or give you specific citations if you were to shoot me an email, which you can get me at mulligan@illinoisstate.edu.
All right. Thank you so much to all of our presenters. We're going to switch everyone's layout now to our closing. As Artem has mentioned there in the chat pod, you will see in the bottom left-hand corner of your screen there is a link, a direct download link to all of today's presentations. They are all PDF versions of today's slide decks. So feel free to download those before you leave today. Simply click on the title of that file and then click the download files directly below that, and you'll be able to get those before you leave. Also, we are recording today's webinar, as we have with all 4 of our webinars in this series. So we will be turning this around into our archival version over the next few days, and you'll be able to access not only this webinar but also the other 3 that took place previously in this series. Also, you will see there in the middle of your screen we have our customer satisfaction survey. Simply click on the word here. That will open a new window for you, and will take you to our short customer satisfaction survey. It only takes a couple of minutes to complete, but it does provide valuable feedback to us that we're able to adjust the content as well as how we deliver that content out to you, our forensic community. You will also see that Julian posted some links there in our chat pod for our social media outlets such as Facebook, LinkedIn, and Twitter. We also have our podcast link there. Feel free to click on the podcast link and subscribe to our podcast so you can not only go back and listen to all of the podcasts we've put out over the last 2 years, but also any of our future offerings. We also have our website there. Check out all of our no-cost webinars that we have posted on our website, not only our archival versions of the ones that we've done previously, but also our new listings that we have coming up. We have quite a few webinars coming up over the next couple of months, so be sure to check out everything that we are offering out to you, our forensic science community. Again, thank you so much to all of our presenters today, not only for taking the time to present, but also the countless hours that you've put into your research. The forensic community is greatly appreciated, and we're definitely moving in the right direction as a community as a whole. And also thank you to all of our audience members for joining us today. We hope you have a great afternoon, and we will see you in our next webinar.